Hello, my name is Shelby Harris, and today I will be talking to you about the CRISPR Cas system and its many different uses. To start off with some vocabulary, CRISPR stands for Clustered Regulatory Interspace Short Palindromic Repeats. When you find them in the genome, the CRISPR itself is made up of the repeats and then viral DNA spacers, which forms the CRISPR array. The word Cas stands for CRISPR-associated genes, so the different genes that encode for the different Cas9 enzymes, such as Cas1 or Cas9. Recently, there's a new enzyme that a lab in UC Berkeley made called Cas9X. These genes together will form a CRISPR locus. Before we get into it, you need to know what the bacteriophage is, and it's the virus that attacks the bacteria. And that RNA is a short single-stranded copy of DNA. And then there's a picture of a bacteriophage. To start off with a timeline, in 1987, the strange repeats were first observed in bacterial genomes. Then, all the way in 1993, Francisco Mojica is the first researcher to characterize what is now called as the CRISPR locus. He was the first one to find it and look at it. The term CRISPR wasn't used until 2002, and then all the way into 2005, that same researcher now reports that these sequences matched the bacteriophage genome sequences. So he said, well, the sequences inside the CRISPR array match up with viral genomes. So he said that might be important. So in 2005, the Cas9 protein is discovered in Streptococcus thermophilus. I believe it was discovered at a Danum yogurt facility when people there were working with the bacteria that lives inside your yogurt that you eat. In 2006, they proposed that CRISPR might be a bacterial immune system used to attack the viruses that attack the bacteria. And then in 2007, they tested that by integrating phage DNA into the CRISPR array, and this allowed the bacteria to fight off the next attack. So they took some phage DNA and they put it into the Streptococcus thermophilus bacterium, and when the phage attacks the bacteria the second time around, it was able to fight off the phage. So they said, well, oh, then it took up the DNA and it was able to fend itself against that by using it. And then in 2008, it's discovered that CRISPR can target DNA, which was a big discovery into how it's now became a gene editing system. In 2010, it was found that the CRISPR-9 will cut the targeted DNA, so not only will it target it, but it will also cut it in certain places. In 2011, it's discovered that the CRISPR systems can function in other species, not just bacteria. So we now know that we can use CRISPR in plants, in animals, in humans even. We're not just limited to using it in bacteria. In 2013, there was the first time that CRISPR-Cas9 is successfully adapted for gene editing in eukaryotic cells, and this was done with both human and mice cells in the lab. In 2013, it was also discovered that it can be used for genome editing in plants. And then in 2015, they've made up some laws. You couldn't edit human genomes with this type of thing because it's too easy to do, and they wanted to prevent people making changes to the genome because that's now dangerous because we don't know a lot about this. In 2016, the USDA says that CRISPR-Cas9 edited crops do not have to be regulated as GMOs because you couldn't tell them apart from unedited plants and the lack of foreign DNA in the genome. So this basically says that you can't really tell when things are edited because it's so similar to being unedited, and you don't have to input foreign DNA into the plant to edit it, to force it to do something that the plant usually doesn't do. You just have to edit the plant's genome itself. So it couldn't be called a GMO. And then also in 2016, the National Institute of Health approved that CRISPR gene editing could be used for the first time in human trials, and that the CRISPR-9 technology was used to make CAR T cells that helped them fight the cancer that they had. And so to say what happens when the phage attacks, so how does the CRISPR work? So a phage attacks the bacterium in 5 steps. The first thing it does is that it will attach to the cell. So it will grab on with its legs to the bacterium here. It will then insert its DNA into the host cell. then the host cell will now take that DNA and synthesize the viral proteins to make more viruses inside the host cell. Viruses can't replicate by themselves. They need to use the bacterium to do that. And then it will assemble the viruses, the bacteriophages in this case, inside the cell using its newly synthesized proteins. And then as you get so much built up in here, the cell will lyse open and you'll release the brand new bacteriophages, which will go on now to infect more whole cells. And that's how viruses attack cells, and that's how they kill them. How does the CRISPR protect the bacterium? When the bacterium synthesizes foreign DNA, it will begin to transcribe its CRISPR DNA from the CRISPR locus. It starts coding the Cas protein, so Cas9 in this case, that leads into the CRISPR array. The CRISPR repeats codes for CRISPR RNA, and the spacers that are kept are the DNA sequences from former attacks. So if this bacteria had been attacked by, say, this bacteriophage, it will keep that in a file, and it will come up and it will transcribe those genes in order to attach the CRISPR Cas9 enzyme to it, and it will target the viral DNA now to cut it. And when you cut up DNA, it can't replicate, so it destroys its purpose. That the spacers and the repeats make up what's known as guide RNA, and the guide RNA binds to the Cas9, which is how it will go and find the DNA to cut it up to stop the viral infection, so that the viral DNA can't be transcribed and translated into proteins that would kill the bacterium. So like I said, after the guide RNA is added to the Cas9 protein, the whole molecule travels along the phage's DNA strand, and as it travels along the genome, the Cas9 unravels the phage's DNA and compares it to the viral sequencer DNA. When the match is found, the Cas9 uses its 2 molecular scissors to cut the DNA. So like I said, it will travel along and it will find to where this viral DNA matches. So if it travels, say it's being attacked by this one phage that has a repeat for it, it would travel along and it would find the sequence where it matches, and then it would cut it, and then it would just cut all the places that it matches, and then that would cut apart the DNA, and then you wouldn't have functioning viral DNA anymore. The CRISPR in the lab. Researchers have used CRISPR-Cas9's natural ability to cut DNA in the lab. The CRISPR systems are better than any other genome editing that we've had before. So you had like zinc fingers. You had some others that were really hard to use, and they had like a lot of off-target effects. You couldn't really regulate them. You couldn't really find out what they're going to do when you put it to the cell. But since Cas9 itself is targeting specific DNA sequences, it's more regulated, and you can control where it's going to cut. You're not just releasing, a pair of scissors onto some DNA and just cutting it up in certain places. You're going to be cutting it at certain repeats. Cas9 can be modified to allow researchers to cut and add nucleotides in the genome. So a big thing with this is like this cystic fibrosis disease. So CF is only a single gene mutation. There's only one gene that causes CF. So is it possible that we could target that gene with, say, Cas9, and then we could use a modified version of the Cas9 enzyme to, when we cut that gene out, could we insert a functional non-CF gene into the genome, and then we could virtually just cure that person of CF? Could we be able to do that? And we could also use that to target other single gene mutations like, say, sickle cell anemia and other diseases like that. So CRISPR has a lot of different uses. It has a big role in genetics, has big roles in agriculture. Say if you have a plant mutation that's good, you could go in there and input that mutation in there, then versus putting foreign DNA into a plant to make it, say, resistant to cold. And then you would have to label it as a GMO, and people don't like GMOs. But if you use gene editing, then you don't have to label it as a GMO. Food sciences, with the plants and with yogurt, you could form a better line of bacteria for, say, your yogurt or your cheese that you're making, because those need bacteria, those need mold. You could edit your whatever organism you're using to make your food be better. Beer and wine, you could engineer some bacteria and yeast that could help you with that. That could just be better. That could make your product even better than it was before. And again, with the medicine, so this plays back into the genetics role. So our treating our diseases and stuff like that is it's really probably the biggest frontier of gene editing. It's probably what most people pay attention to when they hear gene editing. They're probably not thinking about corn or your yeast in your beer. They're more thinking about like, oh, we could use this to modify humans, whether for good or the bad. So there is some concerns that arises with the use of CRISPR that arises with the use of any type of genome editing. The first question usually most people ask is it safe? Is it safe to edit your genome? Would you have any like off-target effects afterwards with like the CRISPR-Cas9 enzyme go haywire and essentially like cut in random spots, and then that could disable your DNA, and then of course that cell or even that person could say die, or it could ruin, it could cause more mutations that could cause things to go even worse. It's just there's a lot of like health issues that come with it. When people say when you start a new thing, you have to of course ask first, is it safe? Another thing is, what about some downstream effects of gene editing? So say we edit a gene, would that make that gene more say susceptible to further editing? Would your cell recognize that it's been edited and say go and just like forget about it, or like edit it some more because it's noticed? Would normal cells attack cells that are edited? So say like if you're treating that cystic fibrosis patient, it would be very hard to find a way to treat every single cell in your body all at once. It wouldn't edit all at the same time. It wouldn't have the same effects. So say if we have an edited gene on our body and then we have a non-edited gene beside it, well, not gene but cell, would that unedited cell attack that edited cell? Would your body recognize it and say attack it? Would you go into organ failure because of this? This is something that happens with, say, like transplants. Would your body just pose a natural attack to it because it's different from you? What happens with germline, sperm and egg cell or embryonic editing? So this is really big. So like if you're editing babies, if you're editing like fetuses, then a lot of people are going to get into that because that's just kind of a sensitive topic. It's really, people don't like it really when you mess with fetuses. People don't like it when you mess with those just anyways. So there's a lot of concerns there. What about the ethical concerns? Is it right to edit genomes? So, of course, there's the good side of gene editing. You could cure cystic fibrosis or sickle cell anemia. You can make a better plant crop that could feed a lot of different people that could have. They have gene-edited rice that has more nutrients in it, so people who are starving, they don't have to eat as much rice as they would usually have to eat to get the same level of nutrition. Those are good, but then there's also bad things that you can do with gene editing. You could make people stronger, you can make things better, you could edit, say, different things to attack something. There's a lot of different bad things that you can do with gene editing, and then people are saying, well, does the bad outweigh the good? What are some ethical concerns we can do? We just can't go out and just gene edit people. We have to think about it for a second. That plays into playing God with designer babies. So should we really be making the best babies? Should we really be doing that? There was a researcher in China who just recently gene edited these twin girls to make them resistant to HIV. I haven't read a whole bunch about that, but he's gotten into huge trouble with that because he, of course, just gene edited some babies and they got really mad with him. But of course, it's a bit stepping it. We need to practice this some more before we go straight into editing babies. Use for cosmetic reasons. So, like I said before, you can make people stronger and smarter. Is it really ethical to do that? Is it really necessary to do that? Is it fair? Making perfect people, what if, say, probably the rich nations are going to get this first. So like here in America, if this becomes mainstream, we could make people who are genetically better than people in, say, poor countries like in Africa or Latin America, then there's never been that true term where you could say, well, these people are genetically better than these people. People have said that, but it's never been true. But now with this editing, that can become true. You can have sort of a sense a master race because they're genetically better than some other race out there. And they're saying, is that wrong? That sounds pretty bad. Like, what could you do with that? But really the big question you have to ask, do the pros outweigh the cons? So do the pros of having gene editing to treat diseases and to further scientific research outweigh the cons that maybe someone's gonna make their baby a little bit smarter or make their baby a little bit prettier just to increase its like chances of a happy life when it's born? Do those pros outweigh the cons of gene editing? That's the question that a lot of researchers are asking themselves now. So some more information if you're interested in this. I've listed a few TED Talks and a few biopharmaceutical companies and research labs that use CRISPRs if you want to look them up. Jennifer Donnell, she's a really big name in CRISPR. She uses Caribou Biosciences in the Groundhouse Lab. What you need to know about CRISPR, it's a good talk. How CRISPR lets us edit our DNA is good in teaching you how it happens. The ethical dilemma of designer babies, so that's the cons and the ethical concerns about it. And how gene editing can change an entire species forever. So it's a pretty cool talk. I recommend looking it up. It's where they made malaria-resistant mosquitoes that couldn't get infected with the parasite that causes malaria. And they did some tests on it, and they essentially found that in a few generations of mosquitoes, the malaria could be completely wiped out. And so there's some big concerns like malaria kills a whole bunch of people a year. We should be releasing these mosquitoes, and the people are like, no, we shouldn't do that because we still don't know a lot about gene editing yet. So I recommend looking up those talks if you're interested. And here's some sources to just state my work. So thank you for watching this video. I hope that you really enjoyed it. I hope you learned some about CRISPR.